Okay, I'm not seeing any new people coming into the webinar, so let's get started. Hi, everyone. Thank you for coming to one of our webinars in the 2025 webinar series from our WSUB program. I'm Bree Price. I'm the Extension Coordinator for our program. I just have a few things to say before our speaker gives his presentation. The WSU Honeybees and Pollinators program is a cornerstone of the College of Agricultural, Human, and Natural Resource Sciences, abbreviated CONORS, that is dedicated to fostering resilient ecosystems in Washington and beyond. Our mission intertwines innovative research, community engagement, and education to safeguard pollinators pivotal to our food security and environmental health. In partnership with the Connors Resilient Washington Initiative, we're committed to advancing sustainable practices in pollinator-friendly landscapes and ensuring a flourishing future for agriculture and natural resources. We have 4 more webinars in this webinar series this year. For more information, you can visit our website, bees.wsu.edu, and visit the upcoming events page. There will be time to answer any questions you have after the presentation today. So if you have questions, feel free to type them in the Q&A box anytime during the presentation. After this webinar and before you close your browser, you'll be prompted for a short 5-question education and outreach survey. Your participation in this really helps us tailor our education and outreach to what you're wanting to learn about. Today's speaker is Dr. Ryan Kuesel. Ryan is a postdoctoral researcher at our WSUB program in the Department of Entomology. Originally from Holland, Michigan, he earned his doctorate degree at the University of Kentucky, studying pests of blueberry, raspberry, and blackberry. At WSU, Ryan is now studying commercial honeybee colony health by inspecting colonies from partnering commercial apiaries for signs of disease and monitoring growth or collapse. His current research efforts will help identify when and why European Foulbrood becomes symptomatic. This webinar is titled Pesticide Exposures During Pollination. What insecticides and fungicides are honeybee colonies exposed to when moved for commercial pollination and honey production? How do those exposures affect their health? This webinar will explore the effects of pesticide exposures on colony health and detail patterns in pesticide exposures from one case study where commercial colonies were moved from California almonds, Washington tree fruit, canola holding yards, and honey yards. So Ryan, I'm going to hide my video and mute myself, and you are all set to share your screen.
All right. Thanks so much for the great introduction, Bree, and thanks everyone for being here today to hear this talk on pesticide exposures during pollination. All right. As Bree said, I'll be talking to you today in about 2 parts, with the first part being about a study that we at WSU performed in 2022 and 2023. It was looking at pesticide exposures in commercial migratory colonies of honeybees. I'm really glad to have this opportunity to talk to you all live about this today, because there are many factors that affect honeybees and their health. But pesticide exposures is one of the major causes of high colony losses in America and around the world. So I'll first give you some background leading up to that study. Then I'll show you the data of what pesticides we found. And then I'll wrap up with a little bit about what can be done, what steps can be taken by agricultural workers and farmland owners and managers to protect bees from these chemicals. So pesticides are especially important for migratory commercial bee colonies. And those are colonies that are owned and managed by commercial beekeepers and beekeeping operations that are moved around the country by the truckload in order to pollinate all these key crops that you see here on the left. Most fruit crops need pollination from an insect to form good fruits, have good fruit size, good fruit set, and make the fruit that you want to see at the grocery store. And here in Washington, the key pollination-dependent crops are the apples, blueberries, raspberries, cranberries, cherries, pears, and plums. But seasonally, the first stop for nearly all migratory honeybee colonies each year is down south in the San Joaquin Valley of California, where those bees are trucked down in order to pollinate almonds. in early spring. They move down in January, and then they're there to or before January even, depending on the operation. And they're there to pollinate the almond flowers when they bloom in February. So this chart shows the number of colonies that are needed to pollinate a key crop each year in either California for almonds or Washington for everything else. And while the data is from 2020 or 2021, the demand for bees is approximately the same, with a little bit of upward trend over the years. And this really does serve to show how critical migratory bees are for all of these crops, but especially for almonds, which demand 1.6 million colonies of bees to pollinate the whole crop in early February down there in California. But after all of Washington's bees are done down in almonds and they're moved back up north in late February or early March, they're divided up and moved between a lot of the fruit crops that we grow, the blueberry, apple, cherry, and pear here in Washington. And depending on the timing of that bloom each year, depending on the weather, some of them bloom faster than others, but oftentimes each individual colony can be moved pretty rapidly between two or more of those fruit crops in Washington. So still, the demand for colonies and fruit production really doesn't meet the current supply. You can see that the supply of colonies from those Washington beekeepers and beekeepers who move colonies to Washington is about 88,000, but there really is a much greater demand per this study. And that means that we need to keep those hives that we have healthy and happy so that we can continue to meet current supply. We should do our best, absolute best, to protect our colonies and keep them strong. But one of the major issue that beekeepers face holistically is that colonies die out at a rate that makes it really hard to sustain a large supply of those bee colonies for your operation. And there's only so much that can be achieved by splitting large colonies in half in spring when you have losses like these. This is from 2018 to 2019 with those overwintering losses when they're checked in early 2019, where California in that year lost about 42% of their colonies, and Washington lost about 46% of their colonies. And this is data collected by the Bee Informed Partnership, BIP. Well, there's a lot of reasons that these hives die out, shown by this chart that was investigated by and created by Kelly Kulenek in 2017. Queen failure on the left, where she'll stop laying, or she'll die, or is maybe killed off by her workers for not laying well enough, can kill a hive. Starvation, where the bees have too few flowers in the surrounding landscape and not enough supplemental feed, can kill a hive. Varroa mites, those ectoparasites that feed on the bee pupa and adults, can be a key contributor to hive death, also by vectoring viruses to that colony as well. Nosema is the microsporidian parasite that lives in the bee guts and robs the bees of nutrients while causing them to have persistent diarrhea in the adults. It can also contribute, as well as small hive beetle. It's a little beetle that lives in the colony. It eats the comb, and as larva makes its way through that comb and the stored pollen and honey, while it eats it, that can cause the honey to spoil, leaving behind a trail of slime in the comb that can also cause the colony to abscond from that hive, as well as poor winters, where temperature changes happen too rapidly, or if it's too cold for a prolonged period of time, can certainly kill the hive, especially when that hive is already weakened by these other factors and the bee population is small inside of it. And then pesticide exposures, be them acute, where an individual bee is exposed to a high dose of a pesticide that causes it to immediately die, or chronic, where persistent exposures over time harm the ability of the colony to survive, is a very key contributor to hive death. And all of those, plus many more factors, has led to, unfortunately, one of the worst colony die-offs that we've seen in history this past year, the 2024 season through the winter and into the start of 2025. And this is information collected by these groups, Project 8%, American Beekeeping Foundation, the AHPA, and the Honeybee Health Coalition through a survey of beekeepers showing that this year, hobbyist beekeepers that keep about 1 to 49 colonies lost an average of 51% of their colonies over the course of that 2024 year. Sideliner operations lost an average of 54% of their colonies, and then those commercial beekeepers got hit really hard this year. So folks who have more than 500 colonies lost an average of 62% of their colonies this year. So there really needs to be more work to understand why this happens. So tackling colony loss is critical indeed. And since there's too many colony loss factors to count, we at WSU set our sights on pesticide exposures through the study, because we can make a change for honeybees and all pollinators as a whole through action in the realm of pesticides, especially through research and then education of practitioners like this agricultural worker here, who's air blasting almonds with the fungicide and inadvertently also air blasting these 6 pallets of bees as well in the way. We can make a difference in terms of the pesticide exposure. So when people hear the term fungicides, like this person is applying here, they know that it's a chemical that is used to kill fungal pathogens. And the assumption is and has been for a long time that fungicides are fairly safe for non-fungal organisms like bees and birds and humans. But what we as a whole scientific community are finding out now is that fungicides can indeed cause harm to other organisms, including bees. And for bees, it's likely an effect of harming the microbes that are inside of the bee guts. And those microbes, just like our good gut microbiome in humans, helps them to digest their food. So without that microbiome, it could be causing poor nutritional capture from the food that those bees are eating as larvae and adults. And it also can increase the ability of other pesticides to harm the bees through what's called synergistic effects. So when a bee consumes or comes into contact with 2 or more pesticides at once, the chemistries can interact with one another inside the bee and cause more lethal, more problematic effects in the bee than when those pesticides are tested in the laboratory. And the way that those pesticides are tested and approved for use in agriculture is that it does involve applying the pesticides to bees, and they're applied to the I think the thorax of adult bees, and then those adult bees are observed over time for mortality. So that does not capture those synergistic effects when multiple pesticides at once can cause a different effect. So when the WCB team set our sights on investigating those pesticides, we dug into the background research, and it showed that too little research had been done to examine pesticide exposures in almonds. And since every commercial colony in the US has moved down to pollinate almonds, that's a really critical gap in our knowledge. We also found that too little research has been done on what chemicals a given colony contacts when moved between those multiple pollination events and multiple crops across the season. And then past studies have also not examined how differences in regional pest pressure or climate, as well as the effect of the landscape surrounding that site, affects what pesticides a colony is exposed to. And then we also wanted to include more than just explicit pesticide exposures by measuring bee health, their ability to forage, as well as diseases and parasites, and their symbiotic microbiome within those bees as well. So that's a portion of this project that you won't hear today. So after a short exploratory study in 2021, where the results from blueberry and raspberry sites are shown here, it's pretty clear that bee colonies come into contact with numerous different pesticides. In this case, you can see 15 or 10, between 10 and or between 9 and 15 different chemicals were found in the pollen of these bee colonies in that initial study, and that sometimes the concentration in parts per billion of that exposure can be relatively high as well. In this case, the insecticides that we detected in that initial pilot study were carbaryl, which is highly toxic to bees, with a topical dosage of that will kill 50% of those adult bees of 1 microgram per bee, which is a really, really small dosage, as well as the fungicides, common fungicides, boscalid, captain, and fludioxonil. Carbaryl is often sold under the name 7. Ray Olson at the time was a postdoc with WSU, and now they're a professor here. They designed this experiment and applied for a grant and received that grant to track 6 groups of 24 honeybee colonies from 3 different beekeeping operations across 2 full years while measuring their health and sampling them for pesticide exposures. Each of these 3 partner operations across the 2 years managed their hives as normal and moved their experimental hive groups that we labeled with plastic cards to identify which hive is which. They moved them between at least 4 different locations across the year, and we sampled them at these 4 approximate time points in February to March, when all the hives were in almond, and around April to May, I'll call it the spring treatment time point 2, they were in fruit crops, most often apple or cherry, and occasionally in blueberry. Then in the summer, number 3, they were placed in seed crops, mostly canola, carrot, and onion. Washington produces a lot of seeds to grow those crops, and then canola is produced for oil production. And then in stage 4, around August to September, I'll call it the fall replicate usually. They were either placed in a yard where there's good floral forage for honey production, or in a holding yard where they're being treated and prepared for overwintering. And then they had access to either the natural plants or occasionally the alfalfa and buckwheat that was present in those areas at that time and flowering. All right, so in 2022, we tracked 2 groups of 24 hives that were managed by those operations. So there were 6 groups of those 24 hives. And then the next year, we cut back a little bit and tracked those 6 groups between the 3 operations for 16 hives each. So all in all, it was 144 colonies tracked in 2022, and then a separate 96 colonies in 2023. So here is the rough locations we had for 2022. You can see in black over here in California is the January and February sampling, right in that San Joaquin Valley where all the almond production for the United States is done. It's in Fresno, Tulare, and Mariposa counties. Then in green is the April and May sampling around central Washington. Hives were more spread out across Washington then in Lewis, Yakima, and Grant counties. In blue was the June or July summer sampling in Grant, Franklin, and Whitman counties. And then in yellow is the August sampling, August, September, that fall sampling in Grant, Adams, and Whitman counties. I had approximately the same locations the next year. So again, first off, down in California, April or May back in Washington and in Yakima and Grant counties. June and July was Grant or Spokane counties, and then August, September, the hives were in Grant or Spokane counties. So although we did track the health of these colonies at each location and each time point by measuring how much brood and how much bees were in the colonies, as well as taking note of any diseases and the quality of the brood pattern that the queen was laying, I won't talk about that portion of the study today. Instead, you'll have to come back for our next seminar on July 19th, and I'll talk a little bit about that portion. So instead today, with the focus on pesticide exposure, all of the data that I'll show you is from analyses of the pesticide residuals that we found in stored pollen, aka bee bread. So at each location, for each of the 6 groups across the 4 different time points in the season, we collected a subsample of pollen from 8 colonies each. And the way we did it was we used a drill with a hole 2-inch diameter hole punch and drilled straight through that comb so that we could extract it, put it on ice, and then have it frozen at -20 Celsius, about normal freezer temperature, to preserve those chemicals before we could take it back to the laboratory and scoop each individual um, pocket of bee bread out, and then amalgamate it in a pestle and mortar. So we had samples from each hive at that location, and then we further took a subsample of this pollen and added all 8 hives from each location together to get a full understanding of what pesticides those 8 colonies and hopefully all the colonies of that location were exposed to at that given time point. Also, whenever we found a colony that had died out, we took a sample of pollen from that as well through the same method to see if poisoning could be a reason for those hives dying. Then we shipped off this pollen on ice to an external laboratory, the synergistic pesticide laboratory, who analyzed it chemically for the content of those pesticide residuals within it. And at that location, they test for over 200 different pesticides, including insecticides, fungicides, and herbicides altogether. So really, we could capture as much information about what pesticides are used as possible, and they do target those pesticides that are used commonly in American agriculture. And the reason why we tested pollen for pesticides is because pollen often comes into direct contact with those pesticides as they're being applied to the flowering blooms in order to protect them from fungal disease or from herbivory, from other non-beneficial insects. And then bees are collecting that pollen in order to feed to their larvae, and it's needed to supply the protein needed to progress through those different larval stages until they can pupate and become an adult. So without pollen, the bee's brood can't grow, but when that pollen is contaminated with those toxic pesticides, the larva consuming it can be poisoned directly, or the microbiome of beneficial gut bacteria and fungi in the guts of those larvae can be harmed and once again cause them to have a lower ability to digest their food and get as much nutrients as possible out of their food. So especially when, as is often the case in these large acreage of tree fruit, canola, any of the crops, and almonds, the taxonomic diversity of pollen that's available to those bees is low, meaning that the number of different species of flowering plants available for them to forage from is quite low. So the bees' nutritional needs might already be not being met well, and pesticides in the pollen then can be a real problem that augments that nutritional deficiency. All right, let's dive straight into the data. So first up, we're going to take a look at the fungicides, and this is the 2022 fungicides. So on the bottom axis here are samples from each beekeeper and amalgamation of 8 colonies at that location, group, and time. I'll point them out directly, but when they're appended by a D, that means that is from a dead colony. So we have this chunk of 5 dead colonies here, and then a couple here as well. So what we found for fungicides in that 2022 year is that the bees were exposed to very few fungicides actually when they were in almonds, which is a good outcome from that year. However, in the cherries, apples, and blueberries in the spring sampling 2 point, pretty high levels of fungicides were observed, and quite a few diversity as well. The data from that stage 2 was definitely higher from stage 1, so there were significantly more levels of and diversity of pesticides in that season 2 compared to sampling point 1. So the number of fungicides per sample overall, and the detection frequency across the sample, meaning the sample with those detection that had fungicides detected was higher However, residue levels declined in the stages 2 and 3 comparative to that tree fruit level in number 2. In that stage 2 in 2022, 5 of those samples collected from the dead hives contain the highest level of fungicides residues here as well. So that could be from a number of reasons. It could indicate that maybe that was a cause of the hive decline, but it could also be that the bee bread had dried out. The residues are more concentrated in those samples, and that the bees that were no longer in that hive could not consume that pollen from previous sampling points. So we're not exactly sure if that is an indicative of being cause of the mortality or due to other factors. But when the bees were moved over into the seed crops, that canola, carrots, and onions in summer, that sampling round 3, the fungicide diversity and concentration mostly were decreased significantly compared to that tree fruit sampling period. Then both dose and diversity remained quite low in that fall sampling period, indicating, as we assumed, that when they're in more natural areas collecting honey, or if they're placed in some of those holding yards and getting ready for overwintering, they do not have as much contact with those fungicides, but they could still be foraging from cropland that has fungicides applied. So in this case, the spring had the highest concern of fungicide level. And yeah, here's those 5 colonies that had died out that have the high dosages. So in 2023, interestingly, the most severe residue scenario for fungicides occurred in stage 1, almonds, where those residues in stage 1 was much, much higher, even statistically, than our stage 3 and 4. We concluded that these major differences in fungicide levels between 2023 here and 2022 there for almonds was simply that we collected our pollen in 2022 in the week of February 16th, when almonds were still in bloom and the fungicide spray application is still low at that time. But then later on in 2023, we collected our samples about a week later and captured those fungicide sprays that had been applied in order to protect the start of those almond fruits. Next up, then in stage 2 of the tree fruits and blueberries, the fungicides that we detected were about the same as the year prior, with the same detection frequency, where in 100% of our samples we detected fungicides, and about comparable range for that concentration of below 2 micrograms per gram of pollen. But compared to almonds that year, the concentrations that they were exposed to in this tree fruit were quite a bit lower. So it's about the same between year 1 and year 2, but because of that 1 week later difference in almond sampling, we saw that those bees are getting dosages of fungicides if they're staying in those almonds beyond petal fall. So although the overall fungicide residues had pretty clear seasonal patterns, as in for sampling point 3 in summer and 4 in fall, had very, very low fungicide concentrations in this year. There wasn't really a clear residue pattern that explained the presence of all these different fungicides that we could analyze. The fungicide types were really diverse across the season, and many frequently detected types of fungicides were consistently found, whereas others were sporadic at multiple stages and time of the year. Some of the 4 major ones that we consistently found was boscalid, flupyram, tebuconazole, and trifloxystrobin. Then just like so overall, the story on fungicides is that fungicide exposures in February's almonds is very, very time sensitive. If you have your bees in almonds, you'll want to move them out after petal fall as rapidly as possible to prevent them from contacting some of those fungicides. Still high exposure to numerous chemicals in the spring tree fruit and blueberries occurred. That's a time point where consistently they are coming into high contact with those fungicides. We found low exposure levels then from just a few chemicals in those seed crop and even lower to almost no fungicides found in the pollen in fall during that honey production time of the year. All right, next up, we'll take a look at our herbicide exposure. Herbicide exposures that we detected were really quite low. And so far, there's not very much concern for the impact of herbicides on the health of honeybees or pollinators. And also being found in low doses is an encouraging story here that they're not coming into contact with very many, and they're not very highly toxic at all to bees. The most commonly found herbicide across the year is in pink. It's pendimethalin, and it was found in a majority of samples in both almonds and in tree fruits and blueberries, as well as a little bit in that third seed crop production period. The pendimethalin is a selective herbicide that controls broadleaf weeds, and it's a pre-emergent herbicide that's used to keep the weeds from growing around the trunks of those almond and tree fruit and blueberries. We also found a little bit of norflurazone, which, as far as I know, is also another of that pre-emergent herbicide here in purple. And I think it's similar for atrazine as well, or use it to control the weeds around the crops, keep a clean understory, and prevent nutrient and water loss and pest production in that understory. Then for the summer seed crops, we still detected some of that pendimethalin as well as the atrazine. And the atrazine does kill both broadleaf weeds and the monocotyledonous grassy weeds. And then in 2023, the story is pretty much the same. The major herbicide that we detected was that pre-emergent pendimethalin for understory weed control. And we detected the other pre-emergent norflurazone just once in that third seed crop area. And then as well as diuron that we did not detect in the year before was detected in almonds this year. Overall, the story for herbicides is that we found them in very low concentrations within the bee bread, as well as a low diversity, just a select few of those pre-emergent weed killers for the most part, meaning that we still have a low concern for herbicide impacts in honeybees. All right, for the sake of time, I'll include both of the graphs of insecticides here, and the stories are fairly similar as well. So up top is 2022. These are insecticides that were found inside the bee bread, and then 2023 as well. We had a fairly high diversity of insecticides that were noted, some of which are extremely toxic to bees, like carbaryl here in purple was detected fairly frequently, especially in 2023. And carbaryl is highly toxic to bees, but it's commonly applied after petal fall in some of the tree fruit crops, especially in apple, because it mimics the action of auxins and actually helps thin the number of apples that are being grown by each individual tree so that you get a fewer number of large apples that are more marketable than many, many smaller apples without that auxin application. So that's applied from anywhere from 80% petal fall until the fruits are at 16 millimeters in size. So that is a period of time when these bees may still be in the field. And you'll see a lot of beekeepers will move quite rapidly to get their bees out of these fields before right after petal fall and before these carbaryl and other insecticides are applied. Then in orange, you'll see that we even had to have a break in the y-axis of concentration, meaning how much in milligrams or micrograms per gram was detected. And this buprofezin could be found in really, really high levels, especially in that tree fruit period, as well as a little bit into period 3 and 4, and a little bit in almond before. Buprofezin is an insect growth regulator that primarily affects insect larvae, and it does that by impacting chitin synthesis. Chitin is the main building block of the outer shell, that exoskeleton of all these arthropods and insects. And it works by disrupting the production of that chitin. And without the proper creation or synthesis of that chitin, the larvae, as they are trying to grow, molt their skin, they need to grow the new layer of skin under their old skin. And without the ability to produce chitin, they can't grow that second layer of skin underneath their cuticle. So it actually prevents them from being able to molt, meaning that they can't grow and will not be able to mature to adults. So this, while it is fairly selective, targeting mostly those pests, it is able to affect honeybee larvae. And since it's been found in these high concentrations, we do have concern for that buprofezin and the negative impacts that it can cause on the ability of the bee brood to grow within the colony. Here is a graph of the pounds of that active ingredient applied. And this is across all of the different insecticides. But here you'll see in this light blue color is IGR, the insect growth regulators, with the key one being that buprofezin. It's used in high concentrations in the United States, and we do now have concern that it's affecting these bees. And it is labeled as safe for bees, and that we think is largely due to the fact that they're tested on the adult bees, again, by dabbing this buprofezin and all other insecticides onto the backs of adult bees. The adult bees don't need to produce that chitin as do the larvae and the pupa, because the bees are done growing at the adult stage. So it would be safe for those adult bees, but will have negative impacts for the larvae and the pupa. So next up, we'll look at the level of risk, or the risk quotient, which is a value calculated based on how toxic each chemical is and what dosage the chemical was found in the bee bread in. So a higher bar in terms of risk quotient means that there's more of a risk of harm to those bees. And you'll see that in almonds, in that 2022, when we had very low exposures across the board, there's very, very low risk quotient. And note that this risk quotient, I believe, is only looking at insecticides. There's no risk assessments on those fungicides completed yet. And hopefully more scientific work is being done to look at that. So then here in 2023, this is just almonds. Here in cherries and tree fruit, we had a fairly high risk quotient with some exposures to that carbaryl and let's see, which one is it? Spinatorum or sulfoxaflor being the large contributors to the risk of those hives having damage from these insecticides. And then finally, here's the overall pesticide exposures across the board, showing in blue is the fungicides, where we had fairly high fungicide exposure in the almonds, mostly in that 2023 year. Fairly low herbicide exposure, and then insecticides being most commonly found in that stage 2, as well as a little bit in stage 3. All right, so that's our data from that study. Next up, we'll take a quick look at how to protect bees from the perspective of what pesticide applicators can do. So all in all, bees come into contact with pesticides in a wide variety of ways. We have been talking about through pesticide contact through pollen ingestion within the nest down here in the bottom right, as well as consumption of that pollen or nectar that is contaminated by the adults as well, as they're bringing it back to the hive, and also as they're ingesting it for their own needs. You can also have direct contact, meaning that if the bees are in the field on the flowers when those insecticides or fungicides especially are being applied, or if they're in the understory foraging on some of those weeds, they might come into direct contact with those herbicides that are being applied to knock down the understory weeds. And then contact as well this is mostly for the native bees that will collect mud from the ground or leaf material in order to build their own mostly solitary nests. They can come into contact then too if there's pesticide residues or explicitly the pesticides on those materials. So here you'll see the direct foliar pesticide being applied, and those bees that are in the flowers or native bees in a hanging box can come into direct contact with that foliar pesticide. So this is the case when we saw the air blast sprayer that was applying pesticides directly to the almonds and also directly to the bees. A less visible but equally impactful way, mostly for our native bees, it can be, again, by soaking into the soil from having overspray or pesticide drift, where it's not being applied directly, or some of it is missing its target and instead landing on the ground, or being oversprayed and coming into contact with those flowers that our honeybees, as well as the native bees, can be foraging on as well. And then a more complex type of exposure can occur with some new pesticide chemistries. And those are the systemic pesticides, where they're usually applied to the rootstock, applied to the ground, and the plant uptakes those chemicals and can then push it into the pollen and the nectar that the honeybees can be foraging on. And then since it has sequestered those pesticides into the food that the bees are eating, they're contacting the pesticide through their diet. And then also overspray or runoff on a rainy day can contaminate the water that our honeybees need in order to cool their hive. These honeybees on a very hot day will fly to a source of water and drink the water and bring it back to the nest where they can regurgitate it. And then through evaporation, much like how we sweat, they'll cool down the hive in that manner. So if there is runoff or overspray, then that can be in the water that these bees are drinking. So ways that pesticide effects on bees can be mitigated is avoid spraying treatments when possible to open blooms. This is especially true of the insecticides. The fungicides will be harder to do because a lot of times those fungicides are protecting the flowers or the very early formation of fruits. But even if there's no environmental hazard section, which we'll come back to in a couple slides, it can be beneficial to make these applications either at nighttime when the bees are not foraging, or to close up your hives before you're making that application. So this is a great card that is available, produced by the NAPPC, Western SARE, and the Future Farm, or the FFAR groups. And it's an infographic about a new section of the environmental hazards label on all new pesticides in the United States. If they have an impact on bees, there will be a red diamond on the label with a honeybee depicted inside of it. And it will give you information about when to apply it, and how to best make those applications without impacting the bees. So this will be found if you open the label that'll be on each package. Then some additional steps to mitigate exposure can be seen through this diverse cropping system here. So consider this cherry orchard, where you have two different varieties or cultivars of cherries, one that has bloomed earlier and is now at petal fall, and one that is still in bloom and has active honeybee and native bee colonies in them as well. So there's also surrounding natural habitat where the bees can be foraging on some of those wild flowers. So if you're going to make an application, if you are this applicator, you can give the property manager or ideally the beekeeper that owns these bees a call and let them know that they should close up their hives at nighttime before you make this application here. And this is actually a fairly common situation in central Washington with a lot of fruit production having different timings. So this can be a critical way. Just let that beekeeper know that you're going to be making an application of a pesticide, what pesticide it is, and then they can choose how best to manage their bees in that situation. When making applications, you'll want to keep as large a buffer as possible, ideally more than 20 feet between your applicator, in this case, this tractor-mounted air blast sprayer, and any of the natural habitat that has flowering plants on it. And that can prevent some of the pollen from being contaminated with these pesticides. Oftentimes, there can be thousands of bees working at the edge of a field. You can see some tree fruit here, perhaps cherry, and right on the other side of it is this beautiful patch of wildflowers here. And there'll be bees foraging on that, so give it a wide berth when making that application to not have overspray land on those flowers. Also, of course, be mindful and try to keep a 20 feet or more barrier or buffer period between you and those active colonies as well. So don't do what we saw with the applicator in the first couple slides. Then also consider asking your beekeepers to put their hives in large groups, at least 20 feet away from the crop. So in this case, the left side is a great example of putting the hives in one drop, at least 20 feet away from that bramble crop there. Whereas in this case, these hives have been placed right next to these blueberry plots. There's work out of another laboratory at Washington State that has shown that it is as effective, especially for pollinating blueberries, to put the bees in one drop within the yard as it is to have them individually at each post. So if you have the area and if it'll be out of the way of your equipment, do put them in one drop, and then your applicators will have an easier time making those pesticide applications without directly spraying the bee boxes. And then finally, if there are dense stretches of blooming weeds like clovers or dandelions, you can mow them down with a mower to remove any of those flowers before making that pesticide application. And by mowing them down, there won't be those flowers, which won't attract the bees that will come into contact with the pesticide spray. And then here's the last bit of data from the study that we performed that I'll show you. This is for each sampling period and each group of colonies, the diversity of pollen that was found from that bee bread that we collected. You can see here in the almonds, it's hard genetically to discriminate between the prunuses. The way that we were able to get genetic information basically took it down to the genus level. And almonds and cherries and plums are all in that same genus of prunus. But we can tell that in February, the only prunus species that's blooming is that almond. So a vast, vast majority of the pollen that those bees collected in our samples were from that almond bloom. And they had a little bit of other wild. And green means we weren't able to break it down close enough to even the genus level to know what to call it. But green means that it's wildflowers that may have gotten oversprayed or may have had the herbicides applied to them um, for weed control. Then next up in sampling round 2, you see that the majority of the pollen that was collected from a lot of these was that prunus, so the cherry pollen, as well as some of the malice apple. And you can see that especially in 2023, that second year, a lot of the pollen that was collected in these was from that apple, mixed in with some of that from the cherries as well, really showing that the bees are foraging from both apple and cherry simultaneously because the blocks of plantings are close enough together for them to forage on both. And then wild taraxacum is the genus of dandelion. So dandelions are a very common weed, and it's showing that they really are picking up a lot of pollen from dandelions as well as part of their fuel in that early spring bloom. Season sampling point 3 and 4 has quite a bit of diversity, although we can see mostly they're getting pollen from wildflowers here, and then some from the brassicas, the mustards, and canola here, especially in that 2023 season. And then finally, in the fall, that August and September, or peak of summer, I should say, they're getting a lot of their pollen from the wildflowers in the area, and then quite a bit of it from helianthus, which are the sunflowers, the wild sunflowers that you'll see when you're driving through central Washington, blooming in that late summer. So it really shows that it can be important to mow down those dandelions before making a pesticide application. But if you're not going to apply pesticides, you can leave them and they'll be good food for the bees. Well, so this is the last bit that I have for y'all today. And this is what I was talking about earlier about pesticide labels. There's new language in that pollinator protection box of each newly registered pesticide that will give the toxicity in terms of what microgram per bee will kill 50% of adult bees when they're exposed to that level. In red, the label will say that this product is highly toxic to bees if it is less than that 2 micrograms. It is extremely poisonous or toxic to those bees. Then toxic to bees on the label, I would say this product is toxic to bees, means that lethal dose of 50% is less than 11 micrograms, but more than 2 micrograms, so an intermediate level. And then relatively non-toxic will not have anything on the label at all. If you read the full environmental hazard section of the label and you see no mention of the bees on a relatively new product, then that means that it's fairly safe for the bees, and it would take more than 11 micrograms per bee to have an impact on that bee. And that's an extremely high dosage. So here's an example of that on the label. This is TriStar, an insecticide, and it does say under environmental hazards, this product is toxic to wildlife, this product is toxic to bees. So that means that this product falls in that intermediate range, and will give you the knowledge that you should do not apply this product while the bees or other pollinating insects are actively visiting the treated area. And since it's saying actively visiting the treated area, that means that you can make this application to blooms at nighttime when the bees are not out. And then it'll prevent a lot of harm to those bees in that way. All right, so everything that you've seen data-wise from the experiment that was done by us at WSU is published by GA and us in this Journal of Hazardous Materials. The name of the study is Pesticide Exposure Patterns in Honeybees During Migratory Pollination. There's a lot more information in there that I couldn't cover in the time period today. You can find that at Science Direct here, or scan this QR code to have access to it. But know that this publication is not open access, so it will require that you have access to a university's library or pay to read it. So instead, I can provide you a copy of the paper if you're interested in reading it. Just email me at ryan.kuesel@wsu.edu, and I can send you that PDF. All right. Thanks so much to the USDA Foreign Agriculture Service. They funded this project at about a little over $1 million in 2019. So thanks to the USDA task grant for funding this work. And thanks to all these folks. The three folks who were the primary investigators on it were Brandon Hopkins, Dr. Ray Olson, who wrote the proposal, and then Ge Zhang. I'm giving this talk in Ge's stead. He recently accepted a professor position back in his home country. And then thanks again to Riley Reed, as well as Xia Lu. Riley Reed is a graduate student who helped a lot with the field work. And then Xia Lu at the University of Idaho is a statistician who helped us to perform the statistical analysis. And thanks also to all these folks here as well, who helped to do a lot of the work either on paper, in the laboratory, or in the field. And thanks so much to our partner beekeepers as well. We couldn't have done this without your help. All right. Questions?
Feel free to type your questions in the box. We'll wait a few moments.
Do my best to answer them as best I can, and if I can't, then I'll ask you to shoot me an email, and I can give you a more researched answer.
I guess I could ask a question. Ryan, is your next talk about this study as well, but a different approach to it, or is it a completely different study?
There will be data from this study as well, and that'll look at how the health metrics of the hive, frames of bees, and frames of brood mostly, but also brood pattern, um, and any disease symptoms that we noted were impacted by what crop those bees were in, as well as a little bit about what landscape was around it. So what natural flowers and were thought to be available to the bees.
Okay, wonderful. I'm not seeing any questions in the Q&A box. So the webinar we were just talking about is in July. You can register for it. It's called Landscape-Driven Stressors During Migratory Pollination. Ryan is the speaker again, so you can hear more about this research. Thank you everyone for attending this webinar. Before you close your browser, you'll be prompted for that 5-question survey. Please answer it. It's very valuable. And as a reminder, we have 4 more webinars. Like I was mentioning, Ryan's our next speaker. And we are recording this. I see one of the questions is, will this be recorded? It is being recorded, and we'll be posting it on our YouTube channel within a couple hours whenever the Zoom cloud lets me know it's ready. So thank you all for attending.
Thanks, folks. Have a great rest of your weekend.
Thank you. Bye.